hope you've been enjoying the class so far. The purpose of this lecture is to get you ready for your first computer lab in which you're going to learn how to investigate protein structures. Building the skills required to analyze the amino acid sequence for a protein and then using that information to guide an investigation of that protein structure, these are big learning goals for the class. So these are things we want you to get under your belts. So in this lecture, we're going to lay the groundwork for this. We will also take this opportunity to get across some concepts that are going to be useful later in the class when you do the different projects. We want to be sure that everybody's up to speed on the basics of protein structure and polypeptide folding. It's hard to really get much out of looking at protein structures unless you have a firm foundation in that. So in this video, we're going to review these concepts. So this lecture ends up being kind of a catch-all for lots of different things we need for the class. You can use the timestamps given here to navigate back and forth between different parts of this lecture video. In part one, we're going to start with a review of polypeptide structure and folding. Everybody needs to be comfortable with the concept of what is an amino acid, how do they come together via the peptide bond and a polypeptide, and basic polypeptide folding, alpha helix, beta sheet, loose coil. So you need to be comfortable with this before you start looking at protein structures. This is all information you have learned in introductory biology, so if you're comfortable with this, you can just jump to the next section. In part two, we're going to look at different kinds of protein structure representations. So you've probably all seen ribbon diagrams, but there are different ways of looking at protein structures and they tell you different things. So we want a firm foundation in that, and we'll cover that in part two. Then we'll move on in part three to talking about protein domains, the units by which proteins fold up and have their different types of biological activity. This will be important later in the class for a lot of what we're going to be doing. Then in part four, we're going to introduce a protein called B-Raf. So you're going to be looking at B-Raf in detail in your computer lab. So this part of the lecture video will provide some really important background information to help you understand what you're doing in the computer lab. In introducing B-Raf, we're also going to cover some information on protein kinases and the RAS MAP kinase signal transduction pathway that will be useful later in the class when you do the Western blot project. So again, this lecture is kind of a catch-all. There's lots of concepts coming in here that you're going to use throughout the class. So with that being said, let's get started with a quick review of polypeptide structure and folding. There are 20 different amino acids that make up proteins, but they all have the same base structure. The central alpha carbon is a chiral center in most amino acids. These are amino acids, so they have a carboxylic acid group and an amino group bonded to the alpha carbon. The alpha carbon always has a hydrogen atom, and then the R group that is different in each of the 20 amino acids and gives each their distinct chemical properties. At neutral pH, a free amino acid is charged on the carboxylic acid and amino groups. The acid deprotonates to give a negatively charged carboxylate, and amino groups act as weak bases, picking up an additional proton from solution to give the nitrogen a positive charge. We can describe the 20 different amino acids by the chemistry of their R groups. Some amino acids have nonpolar hydrophobic R groups. Other R groups are polar but not charged. Some carry aromatic rings. And other R groups can carry a charge, although that may depend on the pH. Lysine, arginine, and histidine carry a positive charge. Aspartate and glutamate have carboxylic acids in their R groups and carry a negative charge. All the amino acids have a 3-letter abbreviation and a single-letter code to designate them. When you see a protein sequence, it's typically the single-letter code given for each amino acid in that sequence. You can find this information summarized in the beginning of the lab manual. In proteins, individual amino acids are covalently bonded to form the polypeptide chain. Here we have a dipeptide of 2 amino acids. We can now refer to the amino acids as residues because the peptide has the residual chemistry of the 2 free amino acids. The peptide bond forms between the carbonyl carbon of one amino acid residue and the amino group of the next residue. As we add more amino acids to our peptide, you can see the chemistry of the backbone is consistent, and we will just number the R groups without showing their structure. But most proteins are hundreds of amino acids in length. We won't draw them all here, and we can jump to the end of the polypeptide, which we will say is 200 residues long. Regardless of the length, the polypeptide has a polarity, with a free positively charged amino group at one end and a free negatively charged carboxylate at the other. Therefore, we refer to the amino terminal end, abbreviated as the N-terminus, and the carboxy terminal end, abbreviated as the C-terminus. If we want to refer to a particular amino acid residue in the polypeptide, we indicate the number counting from the N-terminus. If we make residue 4 an alanine, this would be alanine 4. This linear polypeptide is not the protein structure. The polypeptide must go through a complex set of folding steps to give the protein its structure and function. Let's look at basic protein folding, and in doing this we can define the 4 levels of protein structure. We will use a mock protein to illustrate how the polypeptide folds across these 4 levels. The primary structure of a protein is simply the linear sequence of amino acids from N-terminus to C-terminus. We are not showing the individual residues here, and are only seeing what the polypeptide backbone would look like if it were stretched out from end to end. Segments of the polypeptide can enter a lower energy state by forming helices and sheets that are driven by interactions between adjacent residues. This localized folding of the polypeptide is referred to as the secondary structure. The ordered types of secondary structure are alpha helix and beta sheet. These ordered segments are interspersed with unordered, flexible regions of polypeptide referred to as loose coil. Alpha helix and beta sheet are formed by hydrogen bonding between the atoms that make up the polypeptide backbone. The hydrogen bonding makes these structures rigid and inflexible, particularly for alpha helix, which you can think of as stiff rods in the protein. Loose coil forms flexible sections between these rigid segments. While the R groups of the residues in these segments determine if the folding can occur by producing a lower energy state, they do not directly participate in any bonding. Rather, the R groups are pointed outward from the helix or sheet. In this way, R group interactions can occur between distant residues in the polypeptide to drive the next level of folding. This higher level folding of the polypeptide is the tertiary structure. This is the folded state of the protein where its unique structure confers its particular biological function. This superfolding of the polypeptide forms from interactions between R groups. This includes charge interactions, hydrophobic interactions, and sometimes covalent bonds in the form of disulfide linkages between 2 cysteine R groups. Interactions between R groups and the polypeptide backbone can also contribute to the tertiary structure. Some proteins function as a single polypeptide, but the majority interact with other polypeptides that are the same or different to form multi-subunit complexes. Multiple polypeptide subunits coming together to form a higher order complex is the quaternary structure. We looked at the folding of a theoretical protein where we just showed the path of the polypeptide backbone. Now let's look at a real protein to get a feel for different kinds of structural representations and what information they tell you. We will also take this opportunity to introduce you to the protein you will examine in your first computer lab. This is a protein called B-Raf. What we are seeing is actually just part of the full-length protein. This is the kinase domain that has the catalytic function of this enzyme. We will get into the details of B-Raf later, but for now let's just focus on the representation. This is a ribbon diagram that shows the path of the polypeptide backbone, similar to what we used to show the folding of our theoretical protein. You can see segments of alpha helix, and these are strands of the polypeptide that are coming together to form beta sheet. The arrows of the beta strands point towards the C terminus, showing the direction of the polypeptide backbone. A ribbon diagram does not show the R groups of the amino acid residues. It doesn't show any of the space occupied by the atoms of the protein. A ribbon diagram is useful because you can easily see how the polypeptide is folded. What should you look for when examining the ribbon diagram for a protein? Well, how about which end is which? The viewing program you will use in the computer lab will allow you to easily find the N terminus and the C terminus. But then what is this gap in the polypeptide? The missing polypeptide is actually present in the B-Raf protein, but it is missing from the structure representation. We will explain why this is the case in the next video. Let's look at some other ways to show the protein structure. This is a ball and stick representation of the same protein, B-Raf. It shows the position of each atom in the backbone and the R groups. By convention, carbon atoms are colored in gray or black, oxygen is shown in red, nitrogen in blue, and sulfur in yellow. Hydrogen atoms are generally not shown. A ball and stick representation is useful if you want to look at molecular interactions inside the protein or between the protein and a ligand or substrate. Note that it is difficult to follow the path of the polypeptide backbone in this representation. But is this what the protein actually looks like? Keep in mind that we are depicting atoms here, and we can't say what an atom really looks like. They definitely wouldn't be colored in gray, red, and blue. But we can show the space filled by the electron density for each atom. This is a space-fill representation that shows the van der Waals volume for each atom in B-Raf. This is the volume that is the electron density of the atoms. It is difficult to see the protein folding with a space-fill representation, but it gives you a picture of the protein surface. You can see the residues that are exposed to the aqueous environment outside the protein, and you can identify pockets that could potentially bind small molecules. A surface representation more accurately depicts the protein's surface by showing what's called the solvent-accessible surface of the protein. The solvent being water in this case. This can make it easier to find binding pockets. Keep in mind what each type of protein structure representation can tell you. And don't forget that proteins are simply large molecules composed of atoms bonded together to create the polypeptide backbone and R groups. All of the basic principles of chemistry also apply to proteins. Now let's take what we've discussed so far and consider it from a broader perspective. Why do proteins fold up in the way that they do? Every protein has a three-dimensional structure that gives that protein its specific biological function. Does this three-dimensional structure occur randomly from an infinite number of possible structures driven by genetic variation and natural selection for every protein? Every being the key word here? The answer is no. For the most part, proteins fold up following conserved patterns that are found across different proteins and species. These conserved patterns are called domains. Let's consider what domains are and where they come from. Domains are independently folded modules on the polypeptide. Some proteins consist of a single domain, but many have more than one, and some proteins have a handful of different domains on a single polypeptide. Let's look at B-Raf again to see what we mean by this. Previously, we only looked at the catalytic domain of B-Raf, the part that carries out its protein kinase activity. The full-length B-Raf protein is 766 amino acids in length and has 3 different domains. We will talk about what each of these do in a moment. Right now, let's just consider how these 3 domains come together to make the overall structure. This diagram shows the length of the polypeptide and where each domain lies. Domain 3, the kinase domain, is what we looked at in part 2. Here we can see a model of what the 3 domains might look like in the full-length folded tertiary structure of B-Raf. Here is the N-terminus. This is domain 1. Here is domain 2. And here's the kinase domain we looked at earlier, which is domain 3. You can see this is the same structure inside a much longer protein. This is the C-terminus. You can see how each domain is highly ordered and independently folded, and that the polypeptide between them is not folded or ordered in a consistent way. The intervening segment between the domains is loose coil. B-Raf has an exceptionally long loose coil segment in the center of the protein that is intrinsically disordered. This is functionally significant because it means that this protein can position its domains in different configurations where they can interact with each other or other proteins in the cell. This long, flexible segment is referred to as the hinge region. Here we have it stretched out so that it's easier to see the full length of it. Domains 1 and 2 are regulatory units and are part of what controls when the enzyme is off or on for its kinase activity. This sort of beads on a string organization is one common strategy for organizing different functional units in a single polypeptide. This is not the case with all proteins, though. Some proteins are only a single domain. These tend to be smaller proteins without extensive segments of loose coil. Many metabolic enzymes are like this. Other proteins integrate different functional units in their quaternary structure by having single domains on individual polypeptide subunits that come together to form a complex. What else can we say about protein domains? A domain has a biological activity or biochemical function that it gives the protein. These are things like enzyme activity, DNA binding, or the ability to bind another protein. An SH2 domain, for example, binds to a phosphorylated tyrosine residue on another protein. Some domains are purely structural and serve just as building blocks to position other domains on a protein. Let's take a closer look at the 3 domains on our B-Raf model. Domain 3 is the kinase domain. This is the conserved catalytic part of the protein kinase enzyme. This is where the substrates bind and where the enzyme active site is. We will look more carefully at the kinase domain in the last part of the video. Domain 2 is called a C1 or diacylglycerol binding domain. Diacylglycerol is a phospholipid minus the head group, so things that bind to diacylglycerol bind to the cell membrane. Its function is to serve as a membrane anchor for a protein. To be clear, a C1 domain doesn't just bind diacylglycerol, it attaches to the phospholipid bilayer in general. It does this with a loop of nonpolar amino acids that insert into the hydrophobic core of the membrane. Domain 1 binds to another signal transduction protein called RAS. Therefore, this is called a RAS binding domain, or RBD for short. In signal transduction from the cell membrane, RAS binds and activates different effector proteins that propagate the signal. These proteins have a similar RAS binding domain by which they bind to RAS. RAF becomes activated by binding to RAS as part of a cell growth regulatory pathway used by most animal cells. The RAF RBD binds to a specific effector binding site on RAS. You are probably getting the idea that domains are conserved across different proteins. One way of thinking about domains is that they are building blocks that can be assembled in different ways to create proteins with a certain combination of functions. These building blocks don't evolve de novo or from scratch for each protein. Random mutational events can exchange coding DNA between genes to produce new genes that encode proteins with the same domains, or even a new combination of domains. Over time, natural selection can change the amino acid sequence of the domain by selecting for mutations that fine-tune that domain's function. Therefore, a domain will have a similar amino acid sequence and folding pattern in any protein that has that domain. Domains are conserved across the same protein in different organisms, and they're conserved across different proteins with a similar function in a single organism. The kinase domain in B-Raf is conserved amongst different protein kinases. As you will see in your computer lab, a kinase domain has particular structural elements, like the activation loop, that control its catalytic activity. This concludes our discussion of protein domains. In the next part of this video, we will look at what B-Raf does in cells and how its three domains come together to regulate when its protein kinase activity is on or off. It's time to take a more careful look at B-Raf, both what it does in cells and how its activity is regulated. Why so much focus on B-Raf? Well, 3 reasons. You will be looking at the structural details of how B-Raf becomes activated in your first computer lab, so this background information will help you understand what you are looking at. Second, we know that B-Raf is a protein kinase, and protein kinases will come up again later in the class in the sea urchin fertilization signaling project. We want to get some conceptual understanding of protein kinases under our belts now so that we have that for the class in general. Finally, B-Raf is part of a signal transduction pathway that stimulates cell division. The molecular details for how B-Raf's growth-stimulating activity is regulated and how this regulation can become perturbed in cancer cells is a really interesting story. Okay, B-Raf is a protein kinase, and protein kinases feature big in this class, so let's start with that. In general, kinases are a class of enzymes that transfer phosphoryl groups to or from ADP or ATP. Protein kinases specifically transfer a phosphoryl group from ATP onto another protein that is the target or substrate of the kinase. The substrate protein becomes phosphorylated, and this may alter its activity. Protein phosphorylation is a critical mechanism for regulating biological activity in cells. The target protein can become phosphorylated on a serine, threonine, or tyrosine residue. Protein kinases cannot remove the phosphoryl group from a protein. Removing the phosphoryl group requires the activity of a protein phosphatase, which hydrolyzes the bond to release free phosphate. B-Raf is a protein kinase that phosphorylates its target proteins on serine or threonine residues. It is a serine-threonine kinase. This occurs in the context of a well-established signal transduction pathway that stimulates cell division. The RAS MAP kinase pathway is the classic growth stimulatory pathway that is initiated by growth factors binding to the receptors on the cell surface. Parts of this pathway will come into play later in the class, so let's take the time to go through it now. The signal is initiated by some event at the cell surface, typically by some mitogen, something that stimulates cell division, binding to and initiating signal transduction from a transmembrane receptor. A complex of signaling proteins forms at the inside of the plasma membrane. We don't need to worry about all of the details here, but RAS is one of the proteins in this complex. RAS is a class of signaling protein that is called a G protein. This means that it is in its active signaling conformation when it is binding GTP, and that it has a GTPase activity so that it turns itself off. You will learn more about G proteins later in the class. When the signal transduction is occurring, RAS binds to the receptor complex and assumes its active form. This allows it to bind to and activate B-Raf. We will look at the details of this part soon. There is really only one substrate target protein that RAF phosphorylates, and this is another protein kinase called MEK. When B-Raf is bound to RAS and in its active form, it phosphorylates MEK, which turns on MEK's kinase activity. So this is one protein kinase phosphorylating another to turn on its kinase activity so that MEK can go and phosphorylate other proteins. The proteins phosphorylated by MEK are the MAP kinases. That's why this is referred to as the RAS-MAP kinase signal transduction pathway. MAP kinase stands for mitogen activated protein kinase. Yes, even more protein kinases. There are 3 MAP kinases that are phosphorylated by MEK. When you do the fertilization signaling project, you will do a Western blot to look at the phosphorylation of sea urchin MAP kinase. When MEK phosphorylates the MAP kinases, it turns on their catalytic activity, allowing them to phosphorylate a wide variety of different proteins to effect or produce the biological result of all of this biochemical signaling. Although this pathway may signal other effects depending on the cell type, the effect that is most associated with the RAS-MAP kinase pathway is to promote entry into the cell cycle for cell division. This pathway is a primary control point for tissues to stimulate cell growth. In many human cancers, mutations occur that alter the proteins in this pathway so that they become stuck in their active, signal-promoting form. Continual signaling from the RAS-MAP kinase pathway leads to uncontrolled cell division. Mutations to the gene encoding B-Raf are particularly common in human melanoma, the most serious form of skin cancer. Around 60% of melanomas have a mutation at a particular position in the B-Raf gene. This mutation causes B-Raf to be stuck in its active form so that its kinase activity is always on. You will look at how this mutation affects the structure of B-Raf in your computer lab. To help you understand what you are looking at in the computer lab, let's look at how RAF's kinase activity is regulated under normal conditions in the cell. One thing to understand is that there are multiple layers of regulation that control when RAF is in its active or inactive state. This makes sense because of the importance of being able to carefully control the amount of signaling down this critical pathway that causes cells to divide. Let's look at the structural details. Here is B-Raf again, showing it with the hinge region stretched out. The N-terminal part of B-Raf that has the RBD and C1 domains is referred to as the regulatory region. And the kinase domain on the other side of the hinge is the catalytic region. RAF never actually looks like this in cells. In cells that are not dividing and that have the RAS-MAP kinase pathway turned off, RAF assumes an auto-inhibited state, where the hinge region is folded so that the regulatory region makes contact with a kinase domain. The hinge region looks different here so that we can animate it. Here's the regulatory region with the RBD and C1 domains, and here's the kinase domain. RAF actually needs to form a dimer to be in its catalytically active state. This can be a B-Raf homodimer or a heterodimer with another RAF family protein kinase. With the regulatory region folded across the kinase domain, this prevents dimerization and keeps RAF in its inactive state. In this auto-inhibited state, the single B-Raf molecule is free in the cytoplasm and is not bound to the cell membrane. It is complexed with a pair of scaffold proteins called 14-3-3 proteins that are not shown here. Scaffold proteins provide structural support for complexes formed by other proteins. Contact with these scaffold proteins helps to maintain RAF in its autoinhibited state. When the cell receives a growth stimulatory signal, RAS is bound to the receptor complex and goes into its active conformation. The autoinhibited form of RAF can then bind to RAS through its RBD domain. Here we see what this might look like in the cell. The RAF RBD domain makes contact with a specific binding site on RAS. This positions the C1 domain so that it can insert into the cell membrane. Binding of the regulatory region to RAS in the membrane forces it to release the kinase domain. The hinge can then unfold and the kinase domain can extend into the cytoplasm. The kinase domain can now form a dimer with another RAF protein. Here we are only seeing the kinase domain of the second RAF molecule, but it is also a full-length RAF that is tethered to RAS through its flexible hinge region. RAF is now poised for action, but one more thing needs to happen to turn on its catalytic activity so that it can bind ATP and phosphorylate MEK. To see what happens next, we need to zoom into the kinase domain and look at its structure more carefully. Let's introduce two structural elements that are found in the kinase domain, the P loop and the activation loop. These are conserved elements of the kinase domain, so they are found in all protein kinases. The interaction of the P loop and the activation loop is what controls whether the kinase domain is in its active or inactive conformation. Let's take a careful look at the kinase domain and identify these elements. You can see that the kinase domain consists of two lobes, a larger lobe on the C-terminal side and a smaller lobe on the N-terminal side. The ATP binding cleft is between these two lobes. The blue molecule shown here is not ATP, but it is an inhibitor molecule. Its binding overlaps with the ATP binding pocket, so we can use it to see more or less where ATP binds. Here is the activation loop in the C-terminal lobe, and here's the P loop in the N-terminal lobe. When the kinase is in its inactive state, the activation loop is interacting with the P loop in a way that closes off the ATP binding cleft. This structure is the kinase in its inactive state. If we look at where the electron density is for atoms in both loops, you can see that they are touching, and this blocks ATP from coming in between the loops. What sort of interaction is holding the activation loop together with the P loop? You will discover this for yourself when you do the computer lab. But this interaction keeps the kinase in its inactive state. To shift the kinase to its active conformation, two residues on the activation loop become phosphorylated. The phosphorylation disrupts the interaction of the activation loop with the P loop and pushes the activation loop more towards the C-terminal lobe. This opens up the ATP binding cleft. The activation loop movement also positions R groups in the active site so that the kinase reaction can proceed. RAF is now on, and it can phosphorylate its target substrate. This mechanism of activation of the kinase domain is conserved in all protein kinases. You can think of the activation loop as the on-off switch for the kinase. So what phosphorylates the B-Raf activation loop, you might ask? That's a good question. Remember that pulling the regulatory region away from the kinase allowed RAF to form a dimer. Most likely, the activation loop is phosphorylated by the other RAF subunit in the dimer. Although it is possible different protein kinase enzymes may also contribute to phosphorylating the activation loop. Now that you have a feel for B-Raf and how its kinase domain gets activated, you are ready to investigate some of this in more detail in the computer lab. How does the melanoma mutation that activates RAF come into play in all of this? This is what you will look at. This concludes this video, and we'll see you in the lab.